Hi guys. We're going over my notes for life science and cell biology in a nutshell. So we're learning about the nucleus and its contents and the genetic material. So we were going over um, what the nucleus consists of. It has a nucleolus, which is the part of the nucleus that makes um, subunits of the ribosome. Ribosome makes proteins off of our RNA template. And then the nucleus also contains chromatin, which is the dense form of DNA or genetic material that condenses during cell replication into chromosomes, double strands, which are then given to the new forming cells since they replicate and double themselves. And we learned that there's active DNA and uh, inactive DNA. When the cell is not in replication, the DNA is inactive. So this is called heterochromatin. When it's active, it exposes itself for transcription or getting a template of RNA replicated off of itself to make proteins or for proteins off of the sections of DNA called genes. This is euchromatin. So just different terms. So that's about that. That's what the chromosomes look like, and the helix of the DNA. And earlier we learned that DNA is compacted into chromosomes by a 10,000-fold compaction during this division cycle. And when it's loose, it allows the DNA allows itself to be allows itself to be transcribed into RNA for purposes of protein production in the ribosomes. So So this is the inside of a cell, and this is the nucleolus and the nucleus 3D models. Also, we learned that histones, which are proteins, help with the compaction of DNA into the chromatin, which are also these beads structures or compacted structures are nucleosomes. That's what they're called when the DNA is bound by histone proteins. And necessary molecules pass through nuclear pores, and they go through. These molecules are subunits that are necessary for the protein production. They're subunits, nucleotides, etc. And also energy molecules such as ADP, which help with the process of transcription and stuff. So also the layers between the uh, outer layer of the nucleus, which is since the nucleus has a double membrane, there's an area between these membranes connecting with the lumen of the rough endoplasmic reticulum, which is the outer part of the RER. So through the nuclear pores pass building blocks of DNA and RNA, as well as adenosine triphosphate, the energy providing molecule for synthesizing the genetic material. So any proteins that are helping or subunits of proteins, etc., are allowed to pass through the membranes. Double-stranded DNA. Each strand of this molecule, which looks like this, is a series of nucleotides. Each nucleotide is composed of a sugar phosphate compound attached to one of four nitrogen-containing bases. The sugar phosphate compound link together to form the backbone of the strand. Each of the bases strung along the backbone is chemically attracted to corresponding base on the parallel strand of the DNA molecule. This base pairing joins the two strands of the molecule, much as rungs join the two sides of a ladder, and the chemical bonding of the base pairs twists the double strand into a spiral or helical shape. So the base pairs and join the different nucleotides and the nucleotides are a sugar and a phosphate, and while the base pairs are nitrogen containing bases. So the backbone is basically the sides of a ladder. So there's two sides of the ladder. These are sugar phosphate backbone, and in the middle, joining the two sides, are the nitrogen containing base pairs. These join via hydrogen bonds. The bases and the four nucleotide bases are adenine, cytosine, guanine, and thymine. DNA is composed of millions of these bases strung into an apparently limitless variety of sequences. It is in the sequence of bases that the genetic information is contained, each sequence determining the sequence of amino acids to be connected into proteins. A nucleotide sequence sufficient to encode one protein is called a gene. Genes are interspersed along the DNA molecule with other sequences that do not encode proteins. Some of these so-called untranslated regions regulate the activity of the adjacent genes, for example by marking the points at which enzymes begin and cease transcribing the DNA into RNA molecules. Genetic information is encoded in the sequence of the four nucleotide bases. So the sequence determines what kind of proteins are going to be made from those genes, making up the DNA molecule. One of the two DNA strands is transcribed exactly into messenger RNA, with the exception that the thymine base of DNA is replaced by uracil. RNA also contains a slightly different sugar component, ribose, form that of DNA. In DNA, it's deoxyribose in its connecting sugar phosphate chain. Unlike DNA, which is stable throughout the cell's life, um, um, and of which individual strands are passed to cell generations, the RNA is unstable and temporary. We'll continue with this in the next